Hello, I'm Janar Sathananthan. I'm the Chief Medical Officer of Interventional Cardiology at Boston Scientific, and it's a pleasure to join you today with Dr. Eric Secemsky, who's a friend, but more importantly, a leader in the space of interventional cardiology. And Eric, you touch a number of different spaces, including the peripheral space as well. So thank you for taking the time to join us today. And we wanted to get your thoughts and guidance on the whole space of DCBs and where they go from. So, you're an expert in the space in addition to having some really fun socks. But, obviously, the introduction of drug-coated balloons in the United States has been a long time coming. It's been available outside of the US for well over a decade. And we've seen, since AGENT came to market as the first commercial product, just a lot of enthusiasm for the therapy as a whole in the coronary side, and you've had experience obviously on the peripheral side. There's been a lot of enthusiasm, a lot of interest in off-label use, and there needs to be data that comes with that. But let's take it back a step and just tell me, from your perspective as a clinician in the field and as a leader in the space, what do you look for in a drug-coated balloon?
Yeah, it's fun to be in two different realms right now, where we've been doing this in the periphery for a decade, and now we're doing this in the coronary, because we've got a lot of learnings already behind us in the peripheral space. And so Paclitaxel DCBs are the only DCBs we have available in the US for the lower extremity. We learned really quickly a couple things. One, they work really well, really important. Two is the drug is part of the secret sauce for our Paclitaxel DCBs. Paclitaxel is incredibly transferable to the vessel wall. And we see the clinical benefit, to the point that I spent a lot of time in the last 2 years before AGENT came around trying to help my coronary operators and even my own practice shove peripheral DCBs in the coronary arteries. And and we know that that's been off label, now on label with the AGENT DCB. So Paclitaxel has taught us that it's efficacious both in the midterm, up to 1 year. Now we have data out 5+ years in the periphery that the drug is true and real and significantly deliverable to the vessel wall. And we've really learned a little bit about how to use this in optimal situations like vessel prep. Even more, we're using intravascular imaging, and we're taking that knowledge and moving that now into the coronary space.
So what features do you think a DCB has to achieve, or where do we need to go to in the future for the technology?
Yeah, the one thing that we still debate a lot about is how much drug and how long do you need drug in the vessel wall? And if you step back and think about the restenotic cascade from balloon angioplasty, just to remind everyone that, we create luminal gain by microdissections or sometimes macrodissections in the lesion. And so we know that really restenotic cascade starts at the time of the procedure, and it really moves out through at least 90 days. And so we know that the drug is critical to be present in the vessel wall during that period. Yeah. That goes from both that acute inflammatory and thrombotic process from the dissection through that whole kind of reorganization and healing process over that 3-month period to be effective. And so I think that's really important when we talk about DCBs, and then we start introducing new therapeutic options for DCBs, new drugs that are being developed. They really need to work in that period, throughout the period, to be effective.
Great. So you touched on a very important point there, Eric, which is, I think, as new technologies are coming into the market, there's maybe a mixed understanding on the mechanism. And as it relates to this technology, the drug science and particularly how it gets to the tissue, how long it stays, we're learning, we're trying to understand between different types of drugs and mechanisms. And you led some work with some colleagues on a preclinical PK study. Can you tell us a little bit about the study and exactly what you were hoping to look at there?
Yeah, so it's a great opportunity to learn from all the technological advances that happen in drug-coated balloons. And so for the audience, we have always had Paclitaxel DCBs in the US, again, starting in the lower extremity, now with AGENT available in coronary. And Paclitaxel is a great agent because it is lipophilic, so it's easily transferable to the vessel wall. We could get predictable levels of retention into the vessel wall out through that whole restenotic cascade. And the clinical data really continue to support that short-term and now mid and long-term benefit. But, a lot of our drug-coated stents, almost all drug-eluting stents now are limus. So there's been this thought that should we match our drugs? And so a lot of the last few years have been focused on, well, maybe we can get limus on a balloon, but you can't just do it like paclitaxel. You got to really manipulate it. You got to put it, it's not lipophilic, it washes away, so you got to package it in phospholipids to get it to transfer the vessel wall. We're not really sure even how much is necessary to be effective in the vessel wall. So there's a lot of learning. And I think the key here is, unlike paclitaxel, where we feel really strong about how much tissue should be in there or how much drug should be in the tissue wall, sirolimus, we need to really understand on a device-by-device basis, really if it's effectively getting the vessel wall, and then how long is the vessel wall. And each of them are doing this through different properties, nanoparticles, phospholipid coating, whatnot. So for our PK study, we did probably the first exploration where we had a really large group of devices that we tested in animal coronary model to understand just that. Can the limus versus paclitaxel retention levels be measured? And what do they look like over that really restenotic cascade for these animal models? And do they differ by device? And those are really important questions that we haven't had answers for.
Okay, great. So what did you find in your study when you compared all these different drug-coated balloons?
You know, we found something that was pretty consistent with kind of our pre-work or timing when we've seen this on an individual device level is paclitaxel by far and away gives you the most consistent drug in the vessel wall throughout the period. So it's very clear in these animal models that when you use AGENT DCB, you had therapeutic levels of paclitaxel throughout that 90-day period. And it's really key because, again, that's what's going to match the clinical benefit. The sirolimus devices, again, we looked at a couple of them that are in either evaluation right now or available outside the US. And we saw something very similar. First off, most of the limus DCBs really followed the same pattern. So we've seen other data where there seem to be some device differences based on the manufacturer. But in our study that we did, we saw very similar patterns where you got some nice limus uptake, but pretty quick drop off. And you get lower as you get to about 30 and 60 days. And again, the devices didn't vary much between the different properties of limus packaged on the DCB, but more importantly, we saw a very significant drop off compared to paclitaxel.
Okay. So let me take you through the two components of what were some of the key findings from the study that you did, and talk us through what the potential clinical implications of that are. So I think when you compare the two drugs, you found that paclitaxel, with AGENT, had a higher drug delivery initially at the start compared to limus. So what's the implication for that clinically, do you think?
I think, again, we have to kind of put on our thinking caps of how do we match this preclinical data to the clinical data we're starting to see. And again, I think the highest risk of an event occurs on that peri-procedural period. So that's really when you want your drug present. And we've even seen some preclinical data in the periphery that dissections heal better with paclitaxel present. So I think having really good tissue uptake not only sets you up for the long-term success of tissue retention, you need to get a high concentration up front to be sustained, but also it hits a period where probably you're particularly vulnerable in the context of that intervention, the plasty injury and whatnot.
Okay. So, I mean, maybe it's hard to infer, but would you say a higher dose is better, or we have to understand what the specific dose needs to be? I mean, how do you think about that?
Yeah, I mean, I think, it's like, should we turn the speaker up to 11 when it's already at 10? I mean, I think there's probably a threshold effect of how much paclitaxel is necessary. But once you have to get to that threshold of efficacy, and that's really critical up front. And for people not familiar with DCBs, you have to deliver this through equipment and blood. So we know when you track a DCB in the body, you're going to have some systemic loss. And you need to make sure that enough's on the balloon and transfers to the vessel wall in a short amount of time. So that is really key, is we got to get enough to get past the threshold that we know has been shown to be efficacious from coronary stents with polymers in a one-time treatment.
Yeah, okay. So, we talked about the initial dose at the time, but you also made a comment just about the retention, that you noticed a slower drop-off with the drug with AGENT paclitaxel balloons versus the limus balloon. And I think there's always this comparison with stents, which, with the scaffold is able to achieve that. So how do you think about that retention piece of having just a slower drop-off, and what do you think the implications of that are?
Yeah, absolutely. I mean, I think when we started doing some of this preclinical work, a coronary polymer stent was kind of the benchmark threshold, because the nice thing about a polymer on a stent is you get very predictable levels over that 90-day period. So that's kind of the benchmark we're trying to achieve.